So welcome to the second video in this course G02 on genome assembly. Here we're going to look at why we assemble genomic data. So there's a variety of reasons, most of which stem from the efficient way we can evaluate structures genetic for diversity at hypervariable regions or divergent regions of the genome, such as those associated with immune genes in hosts, or many areas of viruses also are very, very diverse, as well as the same is also true in vectors. We also know that reference genomes are really representative of a single genome at a single instance in time, and therefore are inevitably incomplete, especially at regions that are challenging to sequence. We also have existing questions that we all are aware of that we want to investigate key things that are relevant to our research, like how certain outbreaks arose, or indeed the profile of particular hosts that are of interest. If we're looking at viral sequence data, we might be able to want to determine transition events or the movement of particular lineages between different hosts. And indeed, for hosts, in the same way, we want to understand what particular immune gene variants that host has. And we know that we can ascertain mutations more effectively if we use all the information available in reads, and there's quite a lot. And we also want to have this key focus to understand emerging diseases from both the host, vector, and pathogen point perspective. So what is genome assembly? Genome is the conversion of a set of reads containing redundant sequences into the minimal set of consensus sequences to reflecting the diversity in that original set of reads. So we might specify, for our context, for this particular genome, what sequence length is unique, what sequences are unique for this set of reads in our genome. And our central problem here, particularly for host and vector genomes, is repetitiveness. That many regions of the genome repeat multiple times. And indeed, in viruses, we also have similar problems where there are low complexity regions or gene amplifications. If we look broadly across different species, mainly eukaryotes here, we can see that the genome size varies extensively, and so does repetitiveness. That many prokaryote genomes down here are less repetitive, but there are some, particularly plant genomes, that are very, very large and very, very repetitive. And maize is a good example where the majority of its genome is some sort of repeat. Now, those repeats could be functional, but it certainly means that if we're trying to figure out this issue of repetitiveness, that our challenge is more complex. So if we're dealing with host genomes, that might be somewhere out here, our challenges are very, very different compared to viruses, which might be somewhere around here, where the level of repetitiveness is considerably reduced. Nonetheless, we still have the same issues, that we have potential biases in the preparation of each library. We also have to remove sequencing adapters. We have to consider potential contaminants that have entered into our set of reads. We have to tackle this issue of repeats. We have to also consider if we have sufficient coverage, especially for host and vector genomes. We also know that tends to be a 5.3 decline on average after the first few bases in a read, that the quality of the 3' end is less good. And then we have these known issues about homopolymers in repetitive low complexity regions. We also know that there'll be many technical artifacts in our reads that arise from different sources of errors. And there's a nice paper on this for this down here, and where this figure is taken from. So you know that there can be mutagenic agents that affect our sample, including oxidative damage, that the polymerase can make errors that during the cluster generation process. And then this can be amplified further by PCR, and then there can be further errors from other sources depending on the exact protocol that we use. So we want to eliminate these errors and understand the biological variation that's present without introducing technical variation. There's been some work on the error rate, observed error rate in different devices, and this is after QC and all the usual things that you would do. You can see there for older technologies like MySeq is that the error profile for different studies, each dot shows different studies. might be of the order of a 0.5%. So if we're looking at a mutated site in a read, there's a small chance that it may be an error. And if you look at 200 reads, polymer 2 reads, there's a reasonable expectation if the error is possibly distributed that one base among those 200 is incorrect. So that's why when you actually look at the reads themselves, you'll see a big pileup with random mutations here and there. For newer technologies like HiSeq and NovaSeq and other variations of HiSeq, you can see the error profile is lower. If you zoom in as well, you can see that now you're talking that the error rate might be of 0.1% or thereabouts for newer efforts in sequencing. And that's summarized in this table here. So you can see there for MySeq, the median error rate from those particular studies. And you see then for the new technologies that this is a bit lower, which is good. But there is some variance across all sets. So these are just estimated error rates, and it depends on the reference genome and your whole protocol to generate the sequencing data. So our main goal in this area of genome assembly is to take these input reads, and here we've got paired end reads shown here at the top, all from the same library. We have the sequence tree here, the forward region here, and then we have the reverse region on the other side with some unknown insert size in red intervening between the two. And you can see that the reads are oriented towards each other. And we'll look at that further in G04, we're looking at mutation coding, how we can use that information to quantify the mutation accuracy. And if these reads are all are mapped to the same region, we can merge them into a contig. And same for this contig and same for this contig. And then we can potentially merge these contigs together if there's evidence that they might be joined, say, from a library generated of mate pair reads, MP for mate pair. And that might go from a contig then to a scaffold. For our purposes here, we're not going to deal with scaffolding much, but it is worth bearing in mind that if you're dealing with initial contigs for host and vector genomes, you will want to scaffold them where you may have known gaps of an uncertain length inside your genome sequence. We also are aware that you have long reads and short reads together that contain different types of information. And you should find later on that your long reads assemble very well, they resemble the structure very clearly, whereas the short reads are less good at that. By using multiple technologies together, we kind of get the best of both in terms of the nucleotide level accuracy of short reads and the comprehensive scaffolding and structural resolution from long reads. And that means we can progress from our initial challenge of trying to work out what a picture shows to getting a better resolution and then improving that resolution and try to make it as good as we can progressively. And this, of course, is important that we may have different perspectives on the same data set that tells us different features about them. And as we add in more and more information, we can add on more features, but we also have to be aware that sometimes we find that we have mistaken features that were actually incorrectly identified, and that's why doing learning about genome assembly carefully is really, really important. So a classic way of thinking about genome assembly is the Humpty Dumpty problem. The Humpty Dumpty sat in the wall, Humpty Dumpty had a great fall, all the king's horses and all the king's men couldn't put Humpty Dumpty back together again. And as we know, that's because horses have hooves, so it's going to be quite a difficult challenge. So that's the end of this video. Please view the next video on assembly approaches.